Complex biological systems rely on invisible structural anchors. Within human proteins, specific molecules provide the hydrophobic force required to prevent total architectural collapse. Inside a newborn intensive care unit, a baby just days old presents with progressive lethargy and a highly specific, musty odor lingering in their sweat and urine. While clinicians at the bedside struggle to interpret this rapid neurological decline, the pathology lab down the hall is in full crisis mode. Technicians are firing up mass spectrometers, racing to decode a chemical breakdown that is completely invisible to the naked eye. In medicine, the most catastrophic systemic failures often originate from a single, minute structural anomaly at the atomic level. This schematic isolates L-phenylalanine, an essential, highly hydrophobic amino acid. At a physiological pH, it operates as a net neutral zwitterion, defined by its rigid, flat, water-repelling benzene ring. Because cellular environments are watery, these large, bulky rings are violently repelled by the surrounding fluid. To escape, they are driven deep into the interior of a folding protein through a process called hydrophobic collapse. Once inside, the flat rings physically stack on top of each other. This pi-pi orbital stacking squeezes all remaining water molecules out of the protein's core. This physical act of locking together and banishing the water releases a massive amount of thermodynamic energy. This specific thermodynamic energy provides the permanent molecular glue that allows human cell receptors and metabolic enzymes to maintain their native three-dimensional shapes. Without these tightly packed cornerstones anchoring the architecture, multi-subunit human proteins would lose their shape entirely, making life impossible. A healthy adult carries a total inventory of only 35 to 45 grams of phenylalanine. Paradoxically, even though this molecule supports brain function, 75% of it is locked away inside the actin and myosin chains of our skeletal muscle. The muscle serves as an emergency biological reservoir. During severe trauma or hypercatabolic sepsis, the body breaks down its own muscle tissue, flooding the circulation with phenylalanine to meet immediate bioenergetic demands. The small fraction of phenylalanine that isn't locked in muscle makes its way to the liver. Here, an enzyme called phenylalanine hydroxylase, or PAH, acts as a tightly regulated master valve for the systemic clearing of the molecule. The PAH valve manages blood concentrations by adding a hydroxyl group to the benzene ring, transforming phenylalanine into tyrosine. This provides the chemical foundation for dopamine in the brain, adrenaline for the heart, thyroid hormones, digestive cholecystokinin, and skin melanin. This continuous chain reaction provides the raw chemical materials required to maintain central alertness, vasomotor tone, and metabolic pacing. By managing this conversion, the hepatic PAH valve directly links dietary protein intake to global cognitive drive and circulatory compliance. If a genetic mutation occurs on chromosome 12, an infant is born with a total deficiency of that hepatic PAH enzyme. This missing valve leads to classical phenylketonuria, or PKU. Without the valve to clear it, phenylalanine concentrations skyrocket past the toxic panic threshold of 1,200 micromoles per liter. To enter the brain, molecules must pass through the LAT1 transporter, a specialized revolving door that only accepts large neutral amino acids. When the blood is flooded with toxic levels of phenylalanine, it completely saturates those limited LAT1 turnstiles. This chemical traffic jam competitively blocks other vital amino acids, like tyrosine and tryptophan, from entering the brain. The brain is locked out of its own supply chain. It suffers from central neurotransmitter starvation, halted myelin assembly, and irreversible cognitive impairment. Placental transport loops aggressively pump phenylalanine from the mother into the fetus, reaching toxic concentrations that disrupt embryonic heart and brain development. The neurological damage in PKU results from a transport failure that starves the brain of its essential chemical supply chain. To monitor this tight margin for error, clinical pathology relies on liquid chromatography tandem mass spectrometry, the established standard for measuring amino acids with atomic-level precision. The mass spectrometer separates the molecules, ionizes them, and measures the distinct fragments left behind to confirm their exact chemical identity. Yet this multi-million-dollar technology can be defeated by a single patient variable, the lack of a strict 12-hour fast prior to the blood draw. Drinking a diet soda causes intestinal enzymes to rapidly hydrolyze aspartame, releasing a massive hit of free phenylalanine directly into the bloodstream. The machine will accurately read this toxic spike, causing an analytical fakeout known as pseudo-hyperphenylalanemia, an artifact of the diet, not a genetic liver failure. A second vulnerability occurs during blood collection. If a difficult draw causes red blood cells to burst, a process called mechanical hemolysis, they spill their highly concentrated internal phenylalanine directly into the sample. Despite advanced analytical technology, diagnostic accuracy remains highly susceptible to the physical realities of human digestion and human error at the draw site. To cut through this noise, pathologists evaluate the phenylalanine to tyrosine ratio. Instead of relying on a single raw number, they map the relationship between these two molecules. If both levels rise together, the liver valve is working. But a ratio exceeding 3.0 confirms a broken hepatic valve, separating real classical PKU from a mere dietary fluctuation. Because newborns are neurologically normal at birth, the 7 to 10-day postpartum heel prick strain represents the critical window to save their cognitive trajectory. Because this testing is highly targeted, ordering wide amino acid panels for generalized fatigue without specific metabolic markers is unjustified and creates unnecessary laboratory noise. Understanding this molecule requires aligning 5 core realities: pi-pi thermodynamic stacking, the muscle reservoir, the hepatic PAH valve, LAT1 transporter saturation, and the laboratory traps of fasting and hemolysis. The human genome completely lacks the evolutionary machinery to build this simple benzene ring, yet our focus, our drive, and our survival depend entirely on acquiring it. Our entire cognitive reality is ultimately just the downstream echo of the structural shapes we consume.